Hi, I'm Professor Bolduc, and this is my video on the bacterial cell. I'll begin by describing the different colony and cell morphologies, and then I'll move on to the various cell components that make up bacteria. So let's begin by looking at a few bacterial colonies. Here's just a few examples of the different types of bacteria that can grow on an agar plate that's been exposed to the air. Agar plates like these are used to grow bacteria and sometimes fungus, because they provide a physical support for bacteria to grow on. The developing colonies help us identify the different bacteria that are present in our samples. Each of these colonies began as a single cell. We couldn't see these cells when they first appeared on the agar because of their size, but as they grew, they divided into two cells, which in turn did the same until there were millions of genetically identical cells known as clones, piled one on top of each other, forming these very unique colonies. The differences in the colonies tell us that they're made from different bacteria. Biochemical tests and DNA analyses can tell us the exact genus and species of these bacteria. Now, microbiologists describe each colony by their overall shape, moistness, color, size of diameter, whether they're flat-looking or raised in the center, whether their edges are jagged or smooth, etc., etc. These differences contribute to the bacteria's overall colony morphology. Here's a few more colonies that I've come across in the lab. These colonies came from water samples. These colonies have different colors and textures. The drier colonies, the ones that have less water content, are less shiny, while the moisture ones are shinier and can appear almost mucoid-like. The white colonies in the upper left-hand corner and the bottom yellow colonies are examples of dry, flat colonies with jagged edges. Whereas the top center yellow and the bottom right white and purple colonies are raised or convex in shape, with smoother edges. Both are shiny because they have a higher water content. And colony size is also a unique feature. You'll notice that some bacteria make larger colonies than others. As I mentioned earlier, agar plates provide nutrients. But the problem is that not all bacteria have the same nutritional needs and might even require special growing conditions. Because of this, we can only grow about 1% of all known bacteria in the lab. But hopefully with more research, we'll be able to grow the other 99% of the bacteria. In the meantime, most of what we know about bacteria today is based on these 1%. So now let's look at individual cells. Let's see what they look like. In order to see individual cells, we need to take a sample from a single colony, such as one of these, and smear it on a microscope slide. We'll then add a stain to increase the contrast between the cells and their background and examine them using a compound light microscope. Because of their size, we typically magnify their images 1,000 times. The bacterial smear on the left was stained using a negative staining technique. This technique colors the background, leaving the cells unstained or clear. In a typical stain, you should be able to determine the cell's shape, size, and arrangement. Together, these define the cell's morphology. The majority of the bacteria come in one of 5 different shapes. The small white circles that you see here are individual bacterial cells. Because this smear was made from a single pure colony, all the cells have the same shape and size. The scientific term for spherical cells is coccus. The plural for coccus is cocci. You should use these terms instead of circles or spheres when describing round bacteria. Besides their shape, bacterial cells display specific arrangements. Their arrangement is dependent on whether or not the cells stay attached to each other after cell division. When individual cells are completely separated from each other, their arrangement is said to be that of a single coccus. However, when two cells stay attached to each other, they form what's known as a diplococci. Likewise, when 3 or more are attached to each other in a string-like arrangement, they're called streptococci. When 3 or more cocci form grape-like clusters, they're then called staphylococci. We can say the bacteria shown on the left have an overall arrangement of staphylococci because it's the most prominent arrangement with a few diplococci and streptococci. The ruler in this picture is called a micrometer. Your microscope should come equipped with one to help you size each individual cell. In this picture, 1 ruler division is equal to 1 micrometer. These cocci are approximately 1 to 2 micrometers in diameter. Another typical shape is that of a rod shape. This is called a bacillus. The plural for bacillus is bacilli. Similar to the cocci, bacilli can be found in different arrangements. They can be arranged as single bacillus, diplobacilli, or streptobacilli, but they can never form staphylobacilli. That's because bacilli only attach at their ends, thus they can only form chains of 2 or more cells. Sometimes it might look like you have staphylobacilli type clusters in your field of view, but that's because too many cells were used to make the smear and the cells are simply overcrowded. In this case, a new smear with fewer cells is needed to determine their true arrangement. The bacilli in this picture are a little crowded and can appear in all 3 arrangements. Bacilli do vary in their length and width depending on the species. Some cells have a shape that's found somewhere in between that of a coccus and a bacillus and are hard to differentiate. They actually fall between what we call a coccobacilli. And lastly, there are some bacteria that are elongated with 1 or more curves. The shortest of these are the Vibrio. They look like boomerangs or commas. The mid-sized Spirillum have 2 or more curves and are somewhat rigid compared to that of the larger ones called spirochetes, which are more flexible. The spirochetes and some Spirillum have a corkscrew-like type of helical shape, which is an advantage for them since they can be found in semi-fluid or viscous-like environments. This shape allows them to easily burrow through their environment in search of nutrients. This bacterial smear doesn't contain spirochetes, but I'll show you a moving spirochete in the next couple minutes. In all 3 cases, the curved bacteria only form single-cell arrangements. Their lengths vary, with the Vibrio being the shortest and the spirochetes being the longest. The bacteria in this slide is an example of a Spirillum ranging from 2 to 7 micrometers in length and are very thin, less than a half a micrometer in width. So that pretty much covers the most common cell morphologies that you'll encounter. Now, I'd like to move on and discuss the cellular components found in a bacterial cell. Now, all bacteria are single-cell prokaryotic organisms and are classified as members of the bacteria or the eubacteria domain, which differ from the other single-cell prokaryotes classified in the archaea or the archaeobacteria domain. I'll discuss the archaea in another video. So let's start with the inside of the bacterial cell. As a single-cell organism, bacteria have a jelly-like cytoplasm. The cytoplasm is where all metabolic reactions take place. It contains a lot of proteins, such as enzymes and structural proteins, as well as other macromolecules and water. All these molecules are needed for the cell to grow and replicate itself by binary fission. As a prokaryotic organism, bacteria don't have membrane-bound organelles, nor a nucleus in their cytoplasm. Therefore, all the DNA replication and RNA synthesis takes place inside the cytoplasm. Next, bacteria have a single double-stranded DNA, also known as or also called the chromosome, and they are haploid. They're haploid because they only have 1 copy of the single chromosome. Their DNA is wrapped around histone-like proteins, unlike that of eukaryotic cells that contain histone proteins. Again, unlike eukaryotic chromosomes, bacterial chromosomes are circular, not linear, and their genes are not interrupted with non-coding regions that are called introns. They do, however, contain operons. Operons are a group of genes that are regulated by a common promoter. This is very unique to prokaryotic cells. The region of the cytoplasm that is chromosome dense is called the nucleoid. This term is used to differentiate from the membrane-bound true nucleus that's found in eukaryotic cells. Bacteria may or may not have extra chromosomal DNA called plasmids. But if they do, there could be as few as 1 or 2 copies of the same plasmid, or as many as a couple hundred in the cytoplasm. In any case, they are structurally similar to the chromosome, but much smaller. The genes found in the plasmids are said to be non-essential to the bacteria, meaning that the bacterial cell doesn't need them to grow and replicate. But instead, their genes give the bacteria certain advantages that help them survive and makes them stronger. For an example, the plasmid may contain antibiotic resistance genes that allow the bacteria to survive in an environment that contains that antibiotic. Without the plasmid, the bacteria would die. Next, you'll also find a lot of ribosomes in the cytoplasm. Ribosomes are necessary for protein synthesis. Prokaryotic ribosomes are similar in size to those found in the mitochondria and chloroplasts of eukaryotic cells. They're referred to as 70S ribosomes. The S stands for Svedberg units, after the Swedish chemist Theodor Svedberg, who devised a way to separate cellular particles based on size. These ribosomes are compared to the larger 80S eukaryotic ribosomes found on the surface of the rough endoplasmic reticulum and in the cytoplasm. The fact that prokaryotic ribosomes are similar to those found in the mitochondria and chloroplasts led scientists to develop the endosymbiotic theory, which states that mitochondria and chloroplasts once originated from a symbiotic relationship between 2 separate prokaryotic cells, where one of the cells was internalized and formed the mitochondria or the chloroplast, thus evolving into a eukaryotic cell. Some bacteria also may contain inclusion bodies in their cytoplasms. These are sometimes referred to as elementary bodies. They appear as densely stained pockets inside the cells and can consist of insoluble proteins, carbohydrates, or even gases. The proteins and carbohydrates serve as energy stores or storage, whereas the gases help in buoyancy. Some waterborne photosynthetic bacteria use their gaseous inclusion bodies to float near the surface of the water to allow them to best trap the light for energy. So this completes the internal component part of the bacterial cell, so let's move to the exterior portion of the cell. The cytoplasm itself is surrounded by a cell membrane, which forms the boundary between the cell and its environment. It's also called the plasma membrane or the cytoplasmic membrane. The cell is selectively permeable, which means that it allows certain molecules to enter and/or exit the cell through a process known as simple diffusion. The cell membrane is a phospholipid bilayer. It has 2 layers of phospholipids, similar to that of eukaryotic cells, and it also contains proteins. Some of these proteins span the entirety of the membrane, acting as channels or carrier proteins to help large polar molecules move across the membrane. This is an example of facilitated diffusion. The channel protein known as aquaporins help the overall movement of water, which we know as osmosis, across the membranes. Otherwise, water would have a difficult time crossing the hydrophobic regions of the phospholipids. Unlike eukaryotic cell membranes, the bacterial cell membrane contains hopanoids instead of cholesterol, but they have similar functions that help maintain the cell fluidity. Surrounding the cell membrane is the cell wall, which gives the cell its shape. Make sure you don't confuse the cell wall with the cell membrane. The cell wall also protects the cell from osmolysis. Osmolysis occurs when the cell is in a hypotonic solution, which results in a large amount of water that enters the cell. That's when the water moves in the direction from high water concentration to that of lower water concentration. As it does so, pressure within the cell builds up, pushes the membrane outward and ultimately results in the bursting of the cell. But the mere presence of the cell wall prevents the membrane from overextending itself, thus keeping the cell intact. The major component of the cell wall is called the peptidoglycan. Because it's only found in bacteria, it's a great target for antibacterial therapy. I'll talk more about peptidoglycan and other cell wall components in my other videos. Some bacterial cells have a third layer that surrounds the cell wall, and this is called the Glycocalyx. The Glycocalyx is a matrix of glycoproteins and polysaccharides. Glycoproteins are proteins that are covalently linked to sugars, and polysaccharides are sugars. The glycocalyx can surround the bacterial cell in a loose fashion as shown here, and this is called the slime layer. Or it can be attached in a tighter, more organized manner around the cell, and this is called the capsule. Both the slime layer and the capsule help the bacteria stick to surfaces. An example of bacteria that form a slime layer are the ones in your mouth. Their slime layer helps them stick to your teeth, and as the number of bacteria build up, the bacteria switch to fermentation and produce lactic acid, which eventually destroys your tooth enamel, leading to dental caries. Bacteria that form capsules tend to be more pathogenic. So besides being sticky, capsules prevent macrophage from phagocytizing and killing them, thus allowing the infection to develop into a disease. Besides these three layers, bacterial cells can express different surface appendages. The first one is called the Fimbriae. Fimbriae are short, thin, hair-like structures and are typically found covering the entire surface of the cell. Their tips contain a special protein called an adhesin that helps bacteria bind or adhere to surfaces such as host cell receptors. This is an important step in the process leading to infection and disease. Besides Fimbriae, some bacteria have longer, thicker whip-like structures called flagella. They're structurally different than that of eukaryotic flagella, but have the same function. They're going to help in motility. Some bacteria don't have any flagella, while others can have one or two, or can be completely covered with them. Flagella communicate with special receptors found in the cell membrane that constantly sample the surrounding environment for food, chemicals, and light. This type of movement due to the stimulus is called taxis. The stimulus can be positive, such as the movement towards food, or negative, such as the movement away from chemical toxins. Either way, this is called chemotaxis, movement due to a chemical stimulus, while movement towards sunlight is called phototaxis. The third appendage is called the sex pilus or the F pilus. The pilus is a hollow tube that extends from the cell membrane and fuses with a neighboring bacterial cell if it's close. Bacteria that express a sex pilus have a plasmid that contains the genes needed to form the pilus, along with other genes. The pilus acts as a vehicle for transferring DNA from one bacterial cell to another. As the plasmid replicates, it threads a copy of itself through the pilus into the neighboring cell. This process, known as conjugation, is one of three ways bacteria use to transfer DNA from one cell to another. The other two ways are known as transformation and transduction. I hope to cover these methods in more detail in other videos. Lastly, the Spirillum and spirochete with their unique curved shapes are equipped with a special appendage called the axial filament. Like the flagella, the axial filament is used for motility. In this case, two filaments are anchored at opposite ends. They wrap around the cell and meet midway. The filaments are sandwiched between an outer sheath and a cell membrane. If we were to slice the cell in half and look at the cross-section of the cell, we would see that the filaments form a bulge underneath the sheath. So as the filaments rotate around the length of the cell, the bulge glides across the cell, causing it to turn in a typical corkscrew-type fashion. This type of movement is best for bacteria that live in thick, viscous environments and only works if the cell is helical in shape. Here's a video taken from a water sample. The focus comes in and out, but you can see a long spirochete moving up and down in the water. The cell membrane receptors are sampling the environment for food while the axial filaments are rotating the cell, allowing it to move in either direction, based on the type of the stimulus. Notice its flexibility and that it's approximately 100 micrometers in length, so this makes it much longer than the spirillum. So in summary, I spoke to you about colony morphology, that it can be described by size, color, shape, edges, etc., etc. Whatever term makes the colony unique from other colony types. Then I went on and I mentioned the cell size, shape, and arrangement are used to describe cell morphology. Make sure that you can identify the different bacterial cell shapes as coccus, bacilli, vibrio, etc., and that the cell arrangements are described by adding the prefixes single, diplo, strepto, and staphylo to the words cocci and bacilli. Next, I covered the different cellular components found in prokaryotic cells. Know these differences between those found in prokaryotic cells and eukaryotic cells. Lastly, know the general functions of the different extracellular appendages. This concludes my video on the bacteria.